Genetics is the science of transmission of characters. Genetics primarily deals with the inheritance patterns of characters from the parents to the offsprings. So primarily we understand that genetics is a modern science, and it is a science which deals with heredity and variation. Heredity primarily believes in the characters being transmitted from the parents to the offsprings following a scientific pattern of inheritance. And this pattern of inheritance is termed as classical genetics or transmission genetics. So classical genetics or transmission genetics is otherwise termed as Mendelian genetics. Now the basis of genetics involves the integration of part A, which is termed as transmission or classical genetics, as well as B, molecular genetics, and C, genetics in relation to human welfare. So primarily at our level, we are going to be dealing with relation to transmission genetics. And again, in the first level, we will be looking at what is the genetic basis. That is, what are genes, what are chromosomes, what are the patterns of inheritance, who had studied Mendel's laws, and why is it that we have Pisum sativum as a very important experimental tool in genetics, and what are the 7 contrasting characters of genetics. So coming to concept number 1. The concept number 1 says genetics is the science of inheritance. Genetics is a science of inheritance, and it otherwise deals with heredity. It otherwise deals with heredity. And the meaning of the term inheritance and heredity is related to a very important concept termed as like begets like. That is, mice breed mice, elephants breed elephants, humans breed humans. And genetics also deals with variation. Variation is the variation of characters in terms of evolution. In terms of evolution and time. So the combination of both inheritance as well as variation together is primarily termed as genetics. So genetics is a science of inheritance as well as variation, and it is the basis for evolution. It is the basis for evolution and change. Now in a typical competitive examination, the objective bits primarily ask you what is genetics. So we could always say that it relates to inheritance as well as variation. It deals with heredity, and it is related to the concept of like begets like. The first level of genetics is also termed as transmission genetics or classical genetics. It deals with Mendelian type of inheritance. It forms the basis for evolution in relation to time. So if we want to study classical genetics, if ever we want to study classical genetics, there are several organisms which are very much suited for the study of genetics, like Pisum sativum. Pisum sativum is the garden pea. Then we have even Escherichia coli. Escherichia coli is the bacteria. Parallel to this, we also have another organism called Neurospora crassa. Neurospora crassa. And again we have the mice. So these are all favorable or favorite organisms for study of genetics. The reasons why we have to choose Escherichia coli or Pisum sativum is these organisms are convenient for the study of the transmission of characters. We can easily identify genes from them. We can know the total genome or the total genetic makeup of the particular organism, and it is the basis for molecular biology. Molecular biology is a science of the 21st century which primarily deals with cells, molecules, and the patterns of inheritance. So, while we study genetics, while we are studying classical genetics, a study of Pisum sativum could also be termed as organismal genetics. So, what could be the most favorite organism for study of genetics with relation to plant sciences is Pisum sativum. So, this again is a very important sort of a concept which could easily be coming in any sort of a competitive examination. There could be, while we are studying on Pisum sativum, the term organismal genetics is extremely important. Coming into transmission genetics. Transmission genetics is primarily dealing with the principle of heredity. That is, how characters are transmitted from one generation to the next generation. How are characters transmitted from the parents to the offsprings. And which are all the most important characters. Like you've got somatic characters, you've got genetic characters, as well as you also got neurological characters. And a character is a morphological expression of any particular sort of a trait, like tallness, dwarfness, baldness, intelligence, musical ability, or time of flowering in plants, as well as the character of photosynthetic efficiency or nitrogen fixation which occurs in plants. All these are predominantly termed as characters. And while we study the transmission of characters, it is important that characters are predominantly determined by certain basic units of inheritance which are termed as genes. A gene could, maybe for the classroom, it could be defined as a stretch of DNA which is able to encode, which is able to code for an amino acid as well as for a sort of a protein. So, gene is an unit of inheritance. Many genes are clustered together just like the beads of a string on certain units which are termed as chromosomes. Now, chromosomes are the vehicles of inheritance. Chromosomes are present within any particular nuclei, especially a eukaryotic nuclei. While we take a eukaryotic nuclei, in a eukaryotic nuclei, the chromosomes are well organized. They are well organized just like in the form of spiral rods or spiral coils, and they in turn are the bearers of the hereditary material which is termed as a DNA, which is condensed into genes. So, in transmission genetics, we deal with several levels. It relates to what are the principles of heredity, which we will be shortly learning. Then we will also be looking at the transmission patterns, that is the study of monohybrid as well as the dihybrid cross. And third, we will be looking at how genes are organized and how are chromosomes organized, and later on what is the position of the genes on the chromosomes. So, in short, classical genetics or transmission genetics or Mendelian genetics believes in A. principles of heredity, B. transmission of characters, C. organization of genes and chromosomes, 4. gene mapping or the order and the sequence of the particular genes on the chromosomes.